thanks first of all to Sophion for giving me the opportunity to present our work here today, which will be on the dynamic action potential patch clamp technique, which we use to characterize disease mutations occurring in the voltage-gated sodium channel NaF1.2. So as Daniel already mentioned, I'm working on Metrion Biosciences, which is a contract research organization based in Cambridge, UK, and we focus on preclinical drug discovery, and as such we provide a wide range of services ranging from high throughput fluorescence and automated patch clamp assays, for which we also use Sophion's machines like the QPatch and the Cube, but we also have a big manual patch clamp facility, and we constantly try to develop and incorporate novel techniques into our services. And one of these techniques would be the dynamic action potential patch clamp technique. But before going into details, I would like to give you a little overview of NaF1.2 and its mediated disease spectrum. And at the end, I would like to give you a few examples of such disease mutations we characterized using this technique. And it allows us to give us a better understanding of the physiological consequences of such mutations on neuronal firing compared to conventional voltage clamp assays. And we are in particular interested in better techniques to study such missense mutations because there are currently new variants discovered. And as we heard also today, ion channels in general, they are implicated in a large number of different disorders, and this schematic really only highlights a selected few of these. And such missense mutations, they can occur spontaneously, where they then are called de novo mutations, or they can be inherited. They can be present in the germline or being localized only to a specific tissue, but then they are called somatic mutations. But we still need to functionally characterize these to really see whether they are likely pathogenic. And today, as I said already, I will be focusing on mutations occurring in the SCN2A gene, which encodes the voltage-gated sodium channel NaF1.2. And I know in this audience, I probably don't have to introduce sodium channels a lot. The pore-forming alpha-1 subunit consists of 4 repeated transmembrane domain with the positively charged amino acids responsible for voltage sensing residing in segment 4 of each domain, and then the pore is formed by segments 5 and 6, and they're connecting linkers. The pore-forming alpha-1 subunits, they can also associate with auxiliary beta subunits, which further modify the gating properties and the surface trafficking of these ion channels. Structurally, voltage-gated sodium channels, they form a pseudo-heterotetrameric ion channel with the pore-forming subunits in the center, surrounded by the 4 voltage-sensing domains, which then, upon membrane depolarization, trigger pore opening, allowing sodium to enter the pore. NaF1.2 is expressed widely throughout the CNS. It's located to the axon initial segment, for example, in excitatory cortical neurons, where, especially during early developmental stages, it's responsible for action potential initiation. Later on in development, this role is then taken over by NaF1.6, and NaF1.2 is then thought to play predominantly a role in the propagation, in particular in the back propagation of action potentials. And so far, a large number of mutations have been identified in NaF1.2 in every portion of the channel. And in general, loss of function mutations, shown here in blue, they have been associated with autism spectrum disorders, whereas gain-of-function mutations, shown in red, they have been associated with different forms of epilepsy. However, about a third of these individuals harboring such loss-of-function mutations, they also show epileptic seizures. There also seems to be a correlation between the extent of the induced gating changes and the disease severity, with more moderate gain-of-function mutations associated with benign infantile seizures, which are often inherited. And in these cases, the infants often develop seizures right after birth, but they stop usually after about 2 years of age, and these children have then quite a normal development. However, in the case of more severe gain-of-function mutations, they are associated with infantile epileptic encephalopathy, Also, in this case, the seizures, they start right after birth, but they consist throughout life, and they are also associated with a severe neurodevelopmental disorder. Now, the cessation of the seizures in these more benign variants seems to coincide with the developmental switch to NaV1.6, which has a lower activation threshold and is then recruited to the axon initial segment and then takes over the role of action potential initiation. However, this developmental switch seems not to be able to compensate for these stronger gain-of-function mutations. And now, since we have this diverse disease spectrum with these NaV1.2 missense mutations, this is a further reason to really functionally characterize these variants, also to be able to provide the best therapeutic options for these patients. And I have to say that often these infantile epileptic disorders, they are very treatment resistant, and there are currently efforts being made to identify novel NaV1.2 selective drugs. So usually how such missense mutations are identified, as you well know, is upon recombinant expression in heterologous systems using conventional voltage clamp techniques. But in order to really get a complete overview of all the induced gating effects or changes, one has to apply a variety of different voltage protocols. And this can then be quite time-consuming, both experimentally and in terms of analysis. And very often, such mutations, they are not very clear-cut. They would display features of both gain and loss of function, and this makes it then very hard to interpret what's the physiological consequences of these mutations on neuronal firing. And while recombinant systems, they're obviously very useful tools to study ion channels in isolation, they are easy to manipulate and culture. As I just mentioned, they give limited understanding of the physiological consequences. So for that reason, it would be much better to obviously reconstitute such variants in their native system, like in our case, cultured cortical neurons. But neurons are much harder to work with. They need extended time in culture, and they're also harder to manipulate. And for many ion channels, suitable knockout strains are not even available. And in our case, where we often have to screen quickly tens of mutations, this is just unfortunately not the right system. But in order to circumvent these issues, we use a different approach, a dynamic clamp approach to kind of bridge the gap between these two systems. And dynamic clamp per se is not a very novel technique. It has its origin in cardiac electrophysiology, and it's used to study the effect of simulated computer-generated conductance on an action potential recorded in real time. And these studies are often performed on primary cells like cardiac myocytes or neurons capable of action potential firing. These cells are then recorded in current clamp configuration, and they are coupled to a computer model. Now, the membrane potential of the cell of interest is continuously sampled and fed back into the computer model. The model, as a result, calculates the membrane potential dependent current to be injected back in the myocytes, which then causes an instantaneous change in the membrane potential of the cell, and this is then fed back and changes the model accordingly. But what's important is that these systems, they are in continuous communication with each other, so a change in the cell will affect the model instantaneously and vice versa. And these experiments are often performed to, for example, understand the contribution of conductance to an action potential shape. And here is an example of, in the blue dashed line, this is a native action potential as recorded from a cardiac myocyte. And upon injection of a so-called injury current, which would happen during a cardiac heart attack, this action potential duration is then shortened. Now, for our purpose, we turn this story slightly around. We are now investigating the effect of a real sodium channel conductance on a simulated action potential. And for this reason, we use a computer-generated model of the axon initial segment. And this model has been implemented by the lab of Stephen Petru, who is also the Chief Scientific Officer of Praxis, which are our clients whom we perform this work for, which I'm going to show you later. The model itself is based on the Hodgkin and Huxley formalism to describe the action potential waveform, and it contains a leak conductance, a sodium conductance, and a potassium conductance. Now what we are doing is we are replacing the sodium conductance of the model with our sodium current from our recombinant cell. The model itself is in current clump, so we perform in silico current injection. The model then generates a membrane potential, which is through the amplifier applied to our recombinant cell, expressing our sodium channel variant. And at a certain point, this will elicit a response, sodium current, which is then immediately fed back to the model and will change the membrane potential accordingly. And here is an example of a action potential drain from a wild type NAF 1.2 upon 10 picoampere in silico current injection. But in simple terms, what we are trying to do is to couple a recombinant cell to an in silico neuronal model to kind of create a hybrid neuron. And now I would like to show you a few examples. The first mutation is RQ. Unfortunately, I can't give you any more details about this variant. And here we just performed first some conventional voltage clamp experiments. The variant is shown in magenta, the wild type in black. And as you can see from both the current voltage relationship and the voltage dependence of activation, that the activation is shifted to more negative potentials by about minus 5 millivolts, whereas the voltage dependence of inactivation is unchanged. We also observed slower inactivation kinetics with these variants, as well as an increased persistent current, which would be the fraction of non-inactivating current over a large voltage range. So this variant now shows very clear-cut features of a gain-of-function mutation, and we then applied our dynamic clamp approach on this variant, and as you can see both from the representative traces here and the input-output graph, this variant dramatically increases the firing rate and also shifts the Rayleigh base. So I hope this could convinces you that this dynamic clamp assay is indeed sensitive enough to detect these gating changes and predict an enhanced firing for this variant. And what we are proposing is that it's enough, it can be used instead of a multitude of different voltage clamp protocols to predict the potential physiological consequences. And we looked at an additional mutation, this case LQ, which showed a similar effect on the voltage dependence of activation, which was shifted by about 4.5 millivolts to more negative potentials. Again, the voltage dependence of inactivation was unchanged. Now, in this case, we only saw a marginal change on the inactivation kinetics, and we again had an enhanced persistent current over large voltage range. However, when we now performed our dynamic clamp approach, we saw quite a different firing pattern compared to the previous mutation. It still showed an enhanced firing, but this was much less pronounced. So this now, the reason for this could be because we don't have faster inactivation kinetics, or it could also be due to a gating parameter which we haven't investigated compared to the previous mutation. And so this also shows us that this dynamic clamp approach is also sensitive enough to dissect differences between different variants, so gating differences between different variants. And unfortunately, I did not get to investigate a mutation which showed gain and loss of function features in conventional voltage clamp experiments, which would have been a much more convincing example. But there are published examples by Stephen Petru's group, and they could show that the dynamic clamp then really helped to better predict the physiological consequences. And I hope I could convince you that dynamic action potential clamp is indeed a powerful technique to investigate ion channel mutants. It gives us a better understanding of the physiological consequences of such disease mutations, and it can is also a more direct measure, and it's definitely higher throughput than reconstitution in native system. However, it's a model, and as every model, it has its limitations, and you can also not study any effects on intracellular signaling, which might also be altered with such variants. And unfortunately, up to date, there are only a limited number of such models available. And this brings me to the end of my talk, where I would like to thank Praxis, our clients, and Geza at the Flora Institute, which helped us also setting up the dynamic action potential technique in our lab, everyone at Metrion who contributed to this work, and you for your attention, and please don't hesitate to ask many questions.
Yeah, so maybe I can ask a question. This is fantastic work. I think it's really impressive to see how you can feed that back. How quickly, what is sort of the lag time for the computation to feed back?
So the two systems, it's 140 kilohertz, the communication speed. So okay. It's fast. Okay. We also have a special computer, it doesn't go through normal windows, that wouldn't be fast enough. So we have a special unit which computes it.
Okay, nice. And maybe one more question, because you mentioned at the end, I mean, like what I think, like to just determine the biophysical parameters and then feed it back in a computer model would give you a similar approach. But you mentioned that you found that it's more predictive if you use the dynamic action potential clamp. What do you think is the difference here? Why do you think it's more predictive if you are using the action potential clamp versus just, determining the parameters and feeding it back in the model?
Because if you do, for example, if you do conventional voltage clamp assays, you look at voltage dependence of activation, inactivation kinetics, and often you see a parallel shift of the voltage dependence of activation and inactivation. And then it's quite hard to predict, for example, in terms of neuronal firing, what's going to be the dominant effect. Is it that you will lose a fraction of available channels if the inactivation is shifted to more negative potentials. But at the same time, if the activation is shifted to more inactivation, there might be fire more activated more readily, right? But it really then depends on the resting membrane potential of a given neuron, and it's just hard to guess what's going to be the dominating gating effect. Oops, sorry.
Hi, interesting work. I was wondering, can you predict what happened in the middle between activation and inactivation in channels that are subconductance states? There are, for example, some channels that have like 5 different subconductance states that you can find in the electrophysiology. Can you, can your system predict or profile what is in the middle between activation and inactivation?
I'm not sure I understand your question. Are you relating to window current? No, can you elaborate on your question, please? Because
Yes, as far as I understood your model, you can show in your system in the IV curve and in the IV in the voltage, the activation and inactivation of this particular channel. I'm asking because I'm working with a channel that have 5 subconductance. So it could be 57 picosiemens, it could be 574 picosiemens, and 3 in the middle. So there will be, depending on the stimulus of the channel that I'm working, you will have inactivation or closing on opening states. There won't be like activation or inactivation just like you show in your system. So my question is it possible to predict something in the middle? So, or is it like on, off, and that's all?
So, if I understand correctly, this relates more whether you could understand the underlying gating changes using this model. That probably not, because it will just tell you what's most likely going to be the dominant effect. In that sense, does it make sense? Because
Yes.
yeah.
Yeah. Okay. It's a complicated field. Thank you.
Yeah.